I'm just a guy that wants to cure aging. That's all I am. It's been known for a long time that we all have this theoretical maximum lifespan of 125 years. These are 3 different references where people have looked at what's happened over eons and eons, looking at average ages. We all age because we have multiple sticks of dynamite, where each stick of dynamite is something that's causing us to age. But the big question is, which stick of dynamite has the shortest fuse? I think one of the biggest, shortest fuses is belief. If you don't believe you can live longer, you probably won't. And there's been papers now showing that optimistic people live longer than pessimistic people. Lifestyle choices is another thing that causes short fuse. I mean, if you want to eat at McDonald's every day, you're going to have a short fuse. Biology, that's a very broad subject. I'm going to actually discuss that more on the next slide. But even if we did all that, there's still going to be accidents. Okay, we're still going to fall off cliffs. Let me go back to biology, because that's the main topic that I want to talk about. And we all have biological problems that are going to cause us to age. Genetics. Genetics play a big role. The next one is oxidative stress. I'm going to combine this with mitochondrial dysfunction. A lot of animals die from oxidative stress and mitochondrial dysfunction. And mice are one of the best examples, and all rodents. They suffer tremendously from this. This is why when you put a mouse on a treadmill and run it all the time, it generates a lot of oxidative stress, a lot of free radicals, which damage the cells. It's like a machine gun going off inside the body, just blasting holes in every direction. And this is the main cause of death of old age in mice. What I already explained earlier, that humans are kind of different in that we actually, when we stress ourselves through endurance, we actually boost our antioxidant levels too, so our net oxidative stress is less. But that doesn't mean that's not a problem for us. We also have this stick of dynamite. We are all suffering from oxidative stress and mitochondrial dysfunction. We just need to slow this rate down. Remember, the question is, which stick of dynamite has the shortest fuse? We also have molecular damage, which is constant mutations occurring and stuff like that, and I addressed that a little bit. But that is something else that's causing us to age. We have to figure out how to solve this one. Accumulated junk. Aubrey de Grey talks about this a lot, and the molecular damage. These are things that have to be solved because we are building up a lot of junk in between our cells, maybe a little inside of our cells, and these are causing us to age. Cross-linking, Tony Cerami years ago discovered that we have cross-linking between proteins, and he discovered a receptor on the surface of our protein called the receptor for advanced glycation end products. This research has been going on a long time, but still they haven't got all the solutions. Then lastly, there's cell senescence. No matter if we solved all the other problems, we are still going to have cell senescence that's going to do us in. And so let me go to cell senescence. And the number one cause of cell senescence is telomere shortening. And we have to figure out a way to prevent this. To explain what a telomere is, we have to zoom in on a human being. And we find that a human is made up of 100 trillion cells. And almost all the theories say that we age because our cells age. And we have to zoom in even further. We find that every cell contains a nucleus. And inside these nuclei are found these chromosomes. We've got to zoom in further on one of the chromosomes. A chromosome is contains a long strand of DNA. This DNA is a long string of beads. The beads are called bases. And it's the sequence of these bases that actually give you your hair color, your eye color, and everything like that. I want you to think of this long, long string of beads like a very long shoelace. And at the tip of your shoelaces are caps. These caps protect your shoelaces. Well, our long shoelaces, our long DNA inside our cell, also have caps. They're found at the very tips of our chromosomes, shown in yellow, and these are the telomeres, just like the caps on our shoelaces. Now, this DNA is all wound up like a slinky from one end to the other because it's so big it can't fit inside a chromosome unless it's all condensed into like a slinky. But if we unravel the slinky part of the telomere, we find that a telomere is about 15,000 bases in length. A chromosome is about 100 million bases in length. The telomere is only 15,000 bases in length, at least when we're first conceived. Each and every time our cells divide, our telomeres get a little bit shorter by about 50 to 100 bases. I just want to let you know, what I'm going to explain right now is no longer theory. This is a well-established fact in the scientific community. So as we grow up and our telomeres get shorter and shorter and shorter, when they get down to 5,000 bases, we die of old age. When the telomeres get short, our chromosomes start falling apart. When they start getting shorter, even before they get really short, they're affecting gene expression. But when the telomeres get really short, then the chromosomes start falling apart. Just like your shoelaces start unraveling, our chromosomes do too, except we see it in a way of chromosomes are breaking and rejoining. These are mutations. These are mutations big enough that we can see in the microscope. For every one of these, there's also tremendous numbers of mutations that are too small to see in the microscope. And mutations give us cancer and a lot of diseases. A lot of diseases that we get when we get older are caused by mutations. And this clock, I'm calling it a clock now, because we can measure telomeres and tell people how old they are, how many times their cells have divided. This clock is going at a consistent rate. We can't slow it down below what I call the basal level telomere rate. And after 3 months and 17 days, I missed it by 17 days, and working at Geron Corporation, me and my team discovered this enzyme called human telomerase. The Nobel Prize winners, Elizabeth Blackburn and Carol Greider, and well, at least those two, they had discovered the enzyme activity in 1985 in an organism called Tetrahymena. And so that sounded like, well, this would be a great way, let's see if other animals have this. But nobody could seem to find what this was in humans, because it shows absolutely no relation to what they had discovered in Tetrahymena. When my team discovered this enzyme, it's human telomerase, I'm going to be right up and say this might be one of the biggest medical breakthroughs of all time after we show what it can do. So let me show you. This green thing is a squiggly thing is the DNA, now shown as a double helix. Telomerase is this factory-looking thing. It's bound to the tip of the chromosome, and it's lengthening the telomere. When a cell divides, the telomere gets a little bit shorter, and the clock ticks once. Cell divides again, the telomere gets shorter, and the clock ticks again. This is what's happening inside of our body. But inside of our reproductive cells, this enzyme is produced. And inside of our reproductive cells, and it's the only cell in our body that produces enough of it to actually affect the clock. What happens there is the cell divides, the telomere gets shorter, the clock still ticks, but then telomerase lengthens the telomere and pushes the tick back once. And what we discovered within months after we discovered human telomerase is that if we overproduce telomerase inside of cells, the clock started ticking backward. The telomeres got longer, and it begged the question, did the cells get younger? And in the year 2000, we did experiments where we grew cells from an old person treated with telomerase on the back of a mouse, and the cells were young by every method of measurement you can imagine. If we lengthen the telomeres in us, do we get younger? Do we get cancer? I believe that if we lengthen telomeres, we'll not get cancer. A lot of people do believe this. I believe it's the lack of telomerase that's given us cancer. And that turning on telomerase is going to lengthen our telomeres, prevent those mutations that I was talking about, the rearrangements of our chromosomes, keep our immune system active at fighting cancers if we actually get them, and make it so that things like chemotherapy are going to work better because our cells are not going to adapt to the mutations. I founded Sierra Sciences, and the one and only focus of this company is to find a way to induce telomerase in all of our cells so that we produce enough of it to stop our telomeres from shortening and actually re-lengthen them. This is some pictures from my labs. We have robotic systems. You can see one robot here and another one here. These things test up to 4,000 different chemicals and natural products a day, looking for anything that induces the expression of telomerase. This is some other pictures of the lab. You'll see when we have enough funding, I'm in the lab too. I'm a lab bench scientist. I just want to discuss our strategy because it's actually fairly simple. Let's say a molecular biologist is going to think this is really simple. Maybe other people won't. But I want you to think of, this is a picture of a DNA. It's part of the chromosome. The gray bar is part of our chromosome. And that's where our genes are, like our hair color or eye color. But there's one gene called telomerase. And what that does is produce the telomerase gene. Oh, and next to every gene is a regulatory element. It's oftentimes not right next to it, but there's a regulatory element that turns this gene on and off like a light switch. And in our reproductive cells, that regulatory element is on the on position. Okay. I didn't say this before, but isn't it obvious that these cells can't age? Because if they did age, our children would be born as old, if not older than we are. So even though all the rest of our cells are aging, our reproductive cells don't age. And this is something people have been asking for 50 years. Why is that? Well, it turns out our reproductive cells have no telomere shortening, and it's because of this enzyme telomerase. But they're the only cell in our body that are like this. Every other cell in our body has a protein that binds that regulatory element and shuts it off. That protein is called a repressor. Now, nobody's been able to figure out what that repressor is. There's been a lot of papers published as people have come up with ideas of what that is. Well, in my lab and other labs test it, we can't reproduce the data. We just started taking cells grown in petri dishes, adding chemicals to them one at a time, and asking them 24 hours later, did they start producing telomerase? We have found a lot of these things. We've tested over 400,000 different chemicals now. We have found about 900 that will induce the production of telomerase in normal skin cells. But they're all on the weaker side. I've been in this research now for 22 years. The strongest chemical that we have found to date is something called TAM-818. It's actually called C0314818. It's given that name because that was the 314,818th chemical we looked at. And there were 900 other ones before then, but they were all weaker. So 0 to 100, 100 is what we want to achieve. We actually want to achieve higher than 100. TAM-818 has a score of 16, 16% of the way to finding a drug that will totally stop telomere shortening and hopefully totally stop aging. So some people in New Zealand licensed this from us. They changed the name to TAM-818. They actually made some modifications. They did their own medicinal chemistry to make it even better. But the contract said that we own whatever they discover out of this thing. So it's still our property. And they put it into a skin cream. Okay. Now today, and you've probably heard Molly do the same thing, we're not here to talk about products and stuff like that, so we won't. But what the point is that what we set up these deals is that and we also set up a thing where now a company is selling a natural product for us, okay? That whenever either one of these companies sells those products, we get a royalty. And every penny of that royalty goes into our research. So that's now how we're getting our research done. And we think we're just about, by the end of this year, going to reach a point where we are back to getting the $1 million a month funding that we were used to. The good news is we knew this back in 2008. We know it now. Based on our acceleration of finding hits that were scored at 1%, 3%, 7, 10, 12, 16, we can extrapolate and say, how long will it be before we reach 100? And the medicinal chemists in my company believe they haven't even scratched the surface. They think they can go, maybe even go faster than this. But they think, and I think, that we can reach a score of 100 within 1 year after we get our funding. Thank you very much. My company is Sierra Sciences. We're going to cure aging, or we're going to cure aging.